and Thursday we're taking the exam, and then the following Tuesday there we're still doing this experiment. So last time we talked about kind of the first part of the experiment, which had to do with Le Chatelier's principle. So we're going to talk about sort of the second part of the experiment and sort of the idea of what you need to do, and the calculations and sort of what needs to kind of happen for you to figure out what you're trying to figure out here in part 2 of this experiment. So ultimately, the whole thing is we're looking at the equilibrium constant, really, and a specific equilibrium reaction. And it is the same reaction as what we were dealing with Le Chatelier's principle in part 1. So in this reaction, what we're looking for, or what we're looking at, is the iron 3 ion reacts with the thiocyanate, and it basically forms a complex ion, which is this guy here. And again, the goal here of sort of part 2 of the experiment is to actually find the value of the equilibrium constant. So really, one of the main goals here is to find the value of the equilibrium constant. So if we were to write the equilibrium constant for this guy, it would be the K is equal to the concentration of our product, which is our complex ion, divided by our reactants, which is our iron 3 and our thiocyanate. So again, kind of the main goal here is to actually figure out what this number is. And to do this, we are going to use sort of spectrophotometry, kind of like what we used in the kinetics experiment. Actually, it's the same, should be the same exact sort of equipment that we use in the kinetics experiment. What we're going to do is, as we talked about with the Le Chatelier's part, you know, this side is more of a yellow color. This side is more of a blood red color. And we can use the advantage of sort of the colored solution to help us figure out some important information. And obviously, just a reminder that basically you have a light source. The light source shoots wavelengths of light at different wavelengths through your sample. And then it hits the detector, and we measure absorbance values. And one sort of rule that helps us with absorbance values is Beer's law that we talked about in that sort of kinetics experiment, that A is equal to BC, A being absorbance, C being concentration, and B being Beer's constant. And Beer's constant, excuse me, is basically made up of the molar absorptivity and the path length. So sometimes you'll see people kind of break those up into two different sort of symbols in Beer's law. We just sort of combo them together, which is known as Beer's constant. And in this particular relationship, we could use something like Beer's constant to help us figure out the concentrations of unknowns if we sort of know the absorbance values. And what we're going to do in part 2 of this experiment is, actually, I think it's part 2A, actually. We're going to actually set up a bunch of standard solutions. So we're going to make a bunch of standard solutions. These standard solutions is made up of this reaction, which is the same reaction throughout the entire experiment. And this is going to be done in such a way that they're going to make in part A our thiocyanate really the excess reagent. We're going to use a lot of thiocyanate in there. And we're going to use such an excess of it. What it's essentially going to do is force the equilibrium to go to the right. And what that means is, in the way this was set up, the SCN minus is your excess reagent. That means that by default there, our limiting reagent is going to be our iron 3 in part A of this experiment here. And that's important because by doing this sort of way that this experiment is set up, we're essentially going to force everything to go to the right. And what we know is it is a 1-to-1 relationship between the iron 3 ion and our product. So in part A, when we make these standard curves and we get the absorbance values, we are essentially getting the concentration of our product because we're forcing it basically to 100% head to the right-hand side, and all product's going to be formed. So what's going to happen in part A is there will be a bunch of absorbance values. You'll get absorbance values for how many test tubes? I always want to say there's 5 of them, maybe 5 or 6 test tubes. And you will be able to figure out the concentration of everybody. You'll be able to figure out the iron 3 concentration. You'll be able to figure out the SCN minus concentration from the table. And the one that's really important here is going to be the iron 3 concentration. Again, it is the limiting reagent. It is the one that's going to determine how much product that you make. And in the sense of this first part, the concentration of iron 3 will equal the concentration of our product at equilibrium. So in this first part, the way it's set up, that is the deal. So our kind of initial concentration of our iron 3 will equal the product concentration because of the way this thing was set up. And what you're going to do in part A is essentially make a standard curve where you'll have absorbance, you'll have concentration of your product for those 5 test tubes, 6 test tubes. It hopefully should give you a fairly linear line. You're going to do a best fit line there, maybe better than I could hand draw it there. And it's going to give you an equation of y equals mx plus b. So very similar to what we've done before. We're going to make this standard curve. And this is only for the test tubes that are in part A, as you're going to make this standard curve for. Now, in terms of how you get that information, there's 2 ways you could get that information. When you look at, let me just make sure I find the right one here. I can look at. When you look at the module for this experiment up on Canvas, and let me see if I can find it so I can get the right sort of words there. Okay, so when you look up there on that module, you'll see basically 3 files towards the bottom of the module. And one file will say something like equilibrium of a metal complex ion 1.sp. Then you'll see 2 other files underneath it. One will say equilibrium of a metal complex ion 1 CSV, and it will say set 1 absorbance versus wavelength dot CSV. So just so we're clear, these 2 files here, the ones with the dot CSV, they are to be used in Excel if you want to use Excel. The file up here with the SP, that is to be used with the Pasco software. Both of these files contain the same information. So it's just whichever way you want to analyze these files. You could either use the Pasco software and analyze it through there, or you could use Excel and make it yourself. But in each of these files, either the one Pasco file, you will find both graphs. You will find technically a graph that will look something like this, which is the absorbance versus wavelength, which is this file right here, the Excel equivalent. So this graph that you could find in the Pasco file, once you open it, is this Excel file here. So this is the same equivalent. We'll call it number one, number one. You will also see in the Pasco software, when you open the same file, the one file here, you will see a graph that will come up like this, and they will say something like concentration and absorbance, and there'll be some absorbance values. And this part here, this will be empty. And what that means is you need to do the calculations and figure out the absorbance values. This is for part 2A test tubes. And you need to figure out the concentration of what's important, which is the iron 3, by the way. You need that concentration for those 5 test tubes, and you'll have like 5 absorbance values, and you need to put them in. As you put in the correct concentrations, you will start to see dots being made on the graph, and it will start to be made. And then you could use the Pasco to do the best fit line, the equation, and all that kind of stuff. Now this, which we'll call number 2, is this separate Excel file here. So again, you could do this all in Excel by using these 2 files. You could do both of those things in the Pasco software using this one file. So these files that you see towards the end, these 3 files, this is for basically part 2A, the standard curve part. And again, you don't have to use both. You could either use the Pasco software and do it all, or you could use the Excel software and do it all, or Excel, some type of Excel-like file or software to do it. So your choice. You don't need to do them all. When you open up the Excel files, it will just be numbers, which means you need to go in and make your graphs, and it won't make them for you. The Pascal software, as you do it, this graph will be made for you, and this graph you can make as you're doing it. So that's where you can make your graphs. And in terms of the Pasco software, where you can find those 2 graphs, just to make sure, is when you look at the very top, there should be a box that says Analyze Solution. And then there should be a box next to it that says Concentration, right? And one that says like time, I think, with it as well. So if you click on the Analyze Solution box, it will give you the absorbance versus wavelength graph. So you'll see that graph if you click on that box there. If you click on this box, it will give you that graph of absorbance versus concentration. And again, you'll have a table to the left here that will look something like absorbance and concentration. And again, this part of the table will be filled in for you. So this part will be filled in. This part you need to actually enter. And again, you need to do those calculations. It should walk you through those calculations in the procedure. Question on that there. So the ultimate goal, part 1, is you're going to use that information that's provided to you along with your calculations from the table, and you're going to basically make a standard curve for these standards where iron is your limiting reagent. So the amount of iron that you start with is the same concentration as your product at equilibrium, your complex ions. So again, in part A, that is the way it's set up. Just so we're also clear, when you look at the table, for example, for part A, And again, I believe the test tubes are labeled something like 1A, 2A, 3A, 4A, and like 5A. When you look at that, you'll have kind of 3 columns, I think. I think you'll have a column that is 1 molar, or use whatever it says. I won't put the number just in case it's the wrong concentration. But you'll have KSCN, you'll have HNO3, and you'll have FeNO3. So iron 3 nitrate. And you're going to use this table. Again, you'll have the volumes that you use, like say 5 milliliters, 4 milliliters, 1 milliliter. You're going to use this, and M1V1 equals M2V2 like we've done before to figure out the concentrations of all these guys in the actual mixture when everything's sort of mixed together. Now, a couple of things about this. Obviously, the KSCN is a source of SCN minus. The iron 3 nitrate is a source of iron 3 ion. Nitric acid is used here as a solvent. It is used very much like we use water in a lot of experiments. So in this particular experiment, instead of using water as the solvent, they're using nitric acid as a solvent. So if you remember in the kinetics experiment in those test tubes, they put a certain amount of deionized water in there. Same idea here, the nitric acid is being used as the solvent here. And that's why when you look at something like iron 3 nitrate, when it talks about the iron 3 nitrate solution that you're using, it says something like iron 3 nitrate in 0.1 molar nitric acid. Now, when we're in person, what that means is this solution was made that way. You don't have to add more nitric acid or anything like that. What it is telling you is nitric acid was used here instead of water as the solvent. So it has no effect, the nitric acid's concentration on anything. It is, again, just used as water, basically, or as a solvent in this particular case. So again, when you read that column, it says like iron 3 nitrate in 0.1 molar nitric acid. It basically is just iron 3 nitrate, and iron 3 nitrate is basically just the source of iron 3 ions. So you're going to do this table, figure out the concentrations, I believe. And as we talked about before, this guy in part A will essentially equal the concentration of your complex ion at equilibrium. So they will be the same because of the way it was set up in part A. It's a 1-to-1 relationship. So whatever the initial concentration of iron 3 is will be the equilibrium concentration of your product. And that means when you go to make your Beers plot, what you're really plotting is the concentration of your complex ion versus the absorbance values. Again, going to give you a best fit line where the slope is equal to Beers constant. Any questions on that there? So really part 1 or I guess part A is the way to put that. Part A of number 2 there is to come up with this curve, which is already made for you. Basically, you just got to put it in the concentrations. And the purpose is really to get Beers constant. Any question on that part of it? And the reason why we want this is we're going to do in part 2B, we're going to do this reaction not the same way as they did part A. So what we're going to do here is not make it so that one of them is the limiting reagent and one of them is the excess reagent. We're just going to do this reaction with 7 different test tubes labeled 1B, 2B, 3B, 4B, 5B, 6B, and 7B. And we're going to do this reaction and get the absorbance values for each of these at room temperature. And we're going to get the absorbance value at a higher temperature. So each of you were assigned a data set also in the modules. And I believe it just says equilibrium data set. And you will find the absorbance values for these guys in that file that's called equilibrium data set. So you may have set 1, may have set 2, set 3, set 4, whatever set you have. When you open it, that is what you're going to see. You're going to see absorbance values at a room type temperature, and you're going to see a list of absorbance values at a higher temperature. So for part B, when you open that file, it's a PDF file, you will have a table of, like I said, absorbance values for room temperature for each of these test tubes and absorbance values for a higher temperature for each of these values. And you're going to use those absorbance values and Beer's law to turn those absorbance values into concentrations. So if we resubstitute here for Beer's law, it is the absorbance divided by Beer's constant. This will come from your graph from part 2A. This absorbance value is each of the ones listed here in the table. So that is from the table, or those absorbance values. So you got to do part 2A first to get your Beer's constant. Once you get your Beer's constant, you then can convert your absorbance values that you have for part 2B into concentrations. And what is that going to be the concentration of? It is actually going to be the concentration of your guy at equilibrium. So that is going to be the concentration of your product at equilibrium in part B. So you may be asking yourself, what is all this going to help me do? This is what it's going to help you do. And this is how you would do it by hand. And we'll talk about should you do it by hand in a second, or should you have something else do it for you? And we'll talk about that in just a second. All right, so for example, in part 2B, you go to test tube 1B, for example, right? Test tube 1B. You're going to need to make a ice table for every single test tube that you have for 1B, 2B, 3B, 4B, 5B, 6B, 7B. For each of these test tubes, you will know the initial concentration of both the iron and the thiocyanate. I'm just going to make up the numbers. These are not the right numbers. So I'm just going to say, initially, I'm going to say we started with 2 molar of the iron 3, 4 molar of the SCN minus, and this is going to be 0. You then will go minus X, plus X, which means in my made up example, you would have at equilibrium 2 minus X, 4 minus X, and X. Again, remember that the goal here is to figure out what the value of K is, which is this expression. So we cannot put all these guys in here because we don't know what X is. Right? So this, much like what we did in the very last example today in lecture, we need to kind of work our way backwards. So this is where part A of this comes into play, and what I was just showing you right about here. When you calculate the concentration of your product in each of those test tubes based on the absorbance value, that gives you this guy's concentration at equilibrium. And if you look at our table, this guy at equilibrium equals X. And that equals this guy at equilibrium, which equals the C value you got from the graph in part 2A. So once you know this x value, you know this x value, and you know this x value. So once you figure out that, that will then allow you to actually put the x value in and come up with the equilibrium concentration of iron 3, the equilibrium concentration of thiocyanate, and the equilibrium concentration of your product. And now that you have all these three values, you can actually get a number for each of these guys and put it in. And finally, after all that, you will be able to get a actual number for the equilibrium concentration or equilibrium constant for test tube 1B. The good news and bad news is you then need to do the exact same thing for test tube 2B, 3B, 4B, 5B, 6B, and 7B. Technically speaking, that is 7 ice tables that you need to do for room temperature. And then if that wasn't bad enough, you then need to do the 7 ice tables for the hot temperature. So in case you're keeping track, that's like 14 ice tables if you did them all by hand, which you absolutely could do if you wanted to do them all by hand. I'm not sure I would want to do them all by hand, but if you want to do them all by hand, you could absolutely do it that way. But you can use Excel to help you do that. So if you know how to use Excel or use Excel, you could actually use Excel and formulas and have Excel sort of calculate everything for you, which might be a little bit easier for you to do because you just need to set it up, the calculation once in Excel, and then you could kind of drag it down and it will do all the calculations for you. So I would highly recommend to probably set it up in Excel to do it for you. That usually will require you to have a column in Excel for each of the test tubes numbers. I personally am not familiar with Google Sheets all that much, but I think it works probably similar to Excel. So you probably should be able to put kind of formulas in there. They may be slightly different the way you have to put them in, but it should work probably very similar to Excel would be my guess. Like I said, I'm not actually familiar exactly with Google Sheets and stuff like that. By the way, as well, just so everybody knows, you should have, I believe as a student at Mount SAC, I think somewhere, like if you go to the resource page where it says online resources and all that, I think it has a link where you can get, like, if you need it, Excel, kind of the Office suite and stuff like that, just so you know. I think you can get it for almost free, I believe, if I'm not mistaken, through that link. But Google Sheets or any Apple sort of Excel version or something like that, you know, they all kind of work the same from what I've always seen. But I'm not sure exactly how you put the formulas in specifically for that. But if you're going to do an Excel, you usually will need maybe the test tube number, like 1B, 2B, 3B. You probably will want the initial concentration of your iron 3. You'll probably want a one where you have the initial concentration of your SCN minus. You would probably want a table where you have the equilibrium concentration of your Fe3, maybe one where you have your SCN minus equilibrium. You would also have your complex ion at equilibrium. Sort of table. And remember that this is essentially X in the table. So when you have numbers here, like say 4 and 2, you could say, and this guy it will just say is 1. You basically could tell Excel take equals, you know, we'll call this A1 and AB2. You go, hey, take A1 minus C3, right? And it will give you that number. And then you could do the same sort of thing here. And you could have Excel sort of calculate all those for you, and also have another column where you calculate K, where you basically take those guys and enter it into a formula, dividing those things. So you can use Excel. I would probably recommend doing that because it will probably make it a little bit easier. I would probably do one by hand just to make sure Excel is calculating it correctly, just in case you didn't mess up anything. I would probably spot check one or two of the test tubes by hand to make sure, everything is sort of calculating correctly. But you definitely can use Excel here, or again, an Excel-like program to help you do a lot of these calculations so you don't have to do them by hand. Or I guess if you're bored, you could do them all by hand if you really, really want to. But I definitely would check some out. So the goal here, like I said in part A, is to get that standard curve. Part 2A gets you that standard curve and Beer's constant. Part 2B, you're going to take some unknown solutions, which are labeled as B's, right? These guys are all labeled as A's, test tubes. And you're going to find the K value for, again, a room temperature and a K value for a hot temperature. And you should, when you're done with all these calculations, you should have, I believe, 7 K values, one for each test tube, and perhaps an average K value for room temperature. You also then should have 7 K values for the hot temperature and an average K value for the hot temperature. So when it's all said and done, you should have probably 14 different K values, 7 for room temperature and 7 for hot temperature. You should have an average value for the room temperature K value and an average value for the hot temperature. Couple of things about this, and hopefully what you should see is what we've been talking about. The 7 K values for room temperature, they should be relatively close to each other, right? It is an equilibrium constant. The temperature stays the same. You should get up 7 values that are not identical to each other, but again, should be pretty close to each other. It is still an experiment. Same thing here for the hot temperature. You should end up with 7 K values that are similar to each other, or very close to each other. And when you look at the average value for the room temperature versus the average value for the hot temperature, those two values should be different. Yeah, so they should be different because the temperature is different when you look at those things. Question on sort of the goals here of the experiment. It is a lot of Excel work usually when you do this. And again, like I said, I probably would try to use Excel to get yourself sort of set up and get it to calculate. Because again, you could do all 7 of them really quickly by dragging down, right? As long as you have the right information and the spreadsheet sort of set up correctly. Again, you could do them all by hand if you want to. But I would probably recommend at least doing 1 by hand or 2, and just to double check, make sure Excel is calculating it correctly. When you get these values for K, just so you know, should be a bigger number, I would say. Yeah. So, not really small K value, not super, super large K value, but, higher than 1, I would say, definitely. Any questions on any of that? You will, as we talked about, need to write a formal lab report for this one, and it will need to include a completed ICE table for part 2 here. So again, you'll need to work out one of those as part of your lab report, as that's sort of a sample calculation with actual numbers and stuff like that. Any questions or anything like that? Again, you need to have notebook pages like we talked about with procedure, observations, all that kind of stuff. And you need to work on those things. So again, there's a lot of data analysis and stuff that you have to analyze here. Do not wait to the last minute for sure. Yes. So we're going to work on it today. We have the exam on Thursday, so we're not working on it obviously on Thursday. you could work on it outside of class as well. And then scheduled for next Tuesday, one more day of working on this in class. So again, that should give you plenty of times to do that. Yeah, in terms of the report, you could do one sample ice table kind of from beginning to end is perfect. And like I said, I personally would do that way. And then I would use Excel probably to do all the rest of the calculations for you. Again, if you can't figure out how to do it with Excel, I guess you got to do 14 ice calculations by hand and stuff like that. But in terms of the lab report and showing it, you just need to show one sort of sample calculation. And that's obviously a big calculation here. So one ice table sort of from beginning to end would work probably fine for your report. Other questions? Okay, so remember that you should probably already have the Pasco software downloaded. It's the same one that we use for the kinetics experiment. Again, a reminder that there is one Pasco file. That file contains both the absorbance versus wavelength graph and also the absorbance values for part A, the standard graph. And again, if you're not going to use the Pasco file for that, then you have two separate Excel files that you could use and do the same sort of analysis in Excel. Any other questions on that there? Okay, so, and again, each of you will have a data set, and that data set is for part B of the experiment here. Okay, so for the rest of the time, work on it. If you have questions, make sure that you ask. And I'll open up some breakout rooms in case you want to come out work together, talk it over with each other and that type of stuff.